Our final speaker today is Dr. Helen Zha, an assistant professor in chemical and biological engineering. She serves as an associate director of the New York State Fashion Innovation Center at RPI. Can you believe that? Dr. Zha has received the National Science Foundation Career Award. That's an early investment in the next generation of scientists award. She's also received the National Institute of Health R35 Early Career Maximizing Investigators Research Award, known as the MURI. Helen was named as an emerging investigator by the Royal Society of Chemistry Biomaterials in 2019. She was selected as a SciLog Fellow, which is a group of early career scholars and scientists dedicated to solving global scientific challenges. And in 2023, she received the RPI Class of 1951 Outstanding Teacher Award. Helen's talk today is entitled, "Can Nature Help Us Meet Our Sustainability Goals?" Her research emphasizes how biological systems can meet the sustainability goals while enhancing quality of life. Please help me welcome Helen to the stage.
We've conquered the first challenge, which is I found the mouse. I hope you can all hear me. Okay, it's with great pleasure that I get to talk to you today. So I joined RPI's Chemical Engineering Department in 2018, and this job is amazing. It's afforded me, with the help of my students, this tremendous opportunity to follow my curiosity. So the research in my lab aims to understand how natural materials function, how to develop new platforms for making these materials at scale, and to apply these materials in a broad range of high-impact applications. And in our lab, we work across a variety of fields, everything from sustainable plastics to tissue repair to drug delivery to functional surfaces. So today I'm going to share with you just a snippet of our research. So we like to take material systems from nature as inspiration, and in our lab, we're very inspired by silk. So you may know silk. The Bombyx mori silkworm is probably the one you know best because that's the fiber that you have for your silk clothing. And it's been valued for its strength and toughness and shine for thousands, well, centuries, close to thousands of years in the fashion industry. But other insects also make silk. This is a weaver ant, and its larva makes silk, and the adult weaver ant actually uses its larva as a hot glue gun and moves it across, back and forth, these leaves, these very large leaves that it's using its larva to glue together to make the fortress that the colony lives in. Now, of course, many of you are familiar with spiders, but did you know that one spider web has 6 different kinds of silk? And of course, some of them are sticky, and some of them are extremely good at dampening the energy of incoming prey. Now, interestingly, jumping spiders also make silk, and they don't use these for prey capture. They use them for redirecting their flight midair, and they make silk extremely quickly, about 0.5 meter of silk per second. That's 100 body lengths for the spider a second. And this silk is extremely robust and strong. So how does that help us in our sustainability goals? So I think it's very interesting. Silk can be a really attractive alternative to a lot of the petroleum-based plastics that we use today. For one thing, it's biocompatible. It's already used in sutures. You've probably, if you've ever had sutures, might have had some with silkworm silk in it. And more recently, people have been looking at it as potential edible food packaging to prevent food spoilage. It's flame resistant, which is fantastic for clothing. And for all of you mechies and MSEs out there, here's a stress strain curve or 2. And what you'll see from here is that silk is tougher than Kevlar and about as stiff as nylon. So it's a high performance plastic. On top of that, it can be processed in water, not organic solvents. It's biodegradable, antimicrobial, and it can be renewably made. Now, in my lab, some of the work that we do look to using silk as coatings. On the left here, you can see surfaces on the left side that are, this is stainless steel exposed to certain bacteria, and the bacteria love settling on it and growing on it. And on the right side here, we have our coating on the stainless steel that the bacteria don't want to attach to. In my lab, we've also been using silk coatings to improve the neuroregenerative properties of implants. The bottom strip of green there you see are neurites extended on silk-coated surfaces that are much longer than neurites extended on these plastic surfaces above it. So, silk is amazing, we love it, but how do we get enough silk? If you are scared of spiders, I apologize to you right now, but this is how you might reel or milk a spider for its silk. You anesthetize it and you pin it down and you start pulling from its spinneret and you literally attach it to this little spindle and you start reeling. And this is how you would harvest a silk from a spider. And it's very effective for study, but this is not scalable, right? You can't milk a spider for much before it taps out. And then you would need a lot of spiders, and bad news for you, they eat each other, so you can't keep them in very, very crowded cages. Of course, commercial sericulture is already a thing that exists, but silkworm cultivation has a very negative impact on the environment. It requires large amounts of land and water, and if you're a numbers person, here's some stats to back that up. And at the end of the day, we're still locked into the mechanical properties that this natural material gives us with very little room for tailoring it for the purposes that we might want it for. So we need a method of making a silk or a silk-like material using modern scalable methods. But what is silk? So here's the cool biology/chemistry part. Simply put, silk is a protein, a bit like your hair, but not quite. If you look at a silk fiber, there's a large central core in the middle, that gray area, that contains a class of proteins called spidroins. And if I were to show you an approximation of an amino acid sequence of what a spidroin is like, you see that it looks like it's got these repeating AB amino acid sequence motifs. The A blocks like to form little nanocrystals. The B blocks like to form really stretchy things. And if you are a chemist or a polymer person, you'll say to me, well, that sounds really familiar. That sounds a lot like a thermoplastic elastomer. And I would say yes. Or maybe we should say the thermoplastic elastomer is a lot like silk, right? Which came first? So the closest man-made polymer that we have to silk is something called PBAX, polyether block coamide. You'll often see this as like this cushy material on the bottom of shoes or some kind of sportswear. But interestingly, nature's silk has a combination of toughness and strength that far surpasses any PBAX man has ever made. So nature's got something ahead of us. So what do we do in our lab? So in our lab, I've been working for the past few years, this is in collaboration, by the way, with Professor Matthias Kofas in chemical engineering, to create living systems that can make silk. Our platform of choice is bacteria, which is fast-growing, reaches really high density in liquid culture, so you don't need a big old vat to make a lot of stuff. And it's relatively easy to modify genetically. And so using modern microbiology tools, we engineer various bacteria strains to produce our product of choice, which in this case is silk, which is our protein bioplastic. And so ultimately what I'm saying here is that instead of using petroleum-based chemicals, high temperatures, harsh chemistries to make plastics that are non-biodegradable, we can use living organisms that live in water, require only very modest temperatures, like 30 degrees Celsius, to make high-performance plastics that are naturally biodegradable. And so, don't clap for me. Okay, wait. So when I start describing this idea, many people start to think that I'm talking about some sci-fi Star Trek fantasy future, right? They say, will it really work at scale? And they wonder, and they're right to wonder, because there are a lot of science and engineering challenges that we need to solve to make this reality. But I'd like to point out that we already use living organisms to make many of the commercial products that we use every day. I'm sure some of you appreciated from yesterday or last night that beer is fermented by yeast and produced globally at massive scales in these very large fermentation factories, right? Bioethanol is a fermented product that's been rising in market for the past decades. And we even use cells, mammalian cells, which are quite finicky and complicated to grow, to make our biologics and biopharmaceuticals. So making our plastics using biology, to me, is a reasonable next step. We already have the infrastructure and engineering knowledge in this space of biomanufacturing. We just have to go there, right? So, something interesting that my lab has been working on recently, and this is the particularly cool part that I like about living systems. they're very tolerant to a wide range of food sources that you can give them. So in my group, we asked the question, how can we provide an end-of-life solution to the plastics, that the non-biodegradable waste plastic that we produce by the millions of tons of a year? You could see in the left side here, that's the plastics that we use for packaging, single-use packaging. Incoming is mostly virgin stuff, petrochemical based, and then we use it, and very little of it goes back into the loop by recycling. Most of it gets lost in the environment or landfilled. And how can we take this linear plastic economy, which just generates persistent waste, polyethylene, for example, that will be around for hundreds of years in our environment, and use that as an input into a system where we can create materials that are biodegradable or can be used circularly. So how can we upcycle the waste that comes out of the left side of the system into something sustainable? And of course, I'm going to say microbes to the rescue. So here's how this works. So our collaborators at Argonne National Lab, they took polyethylene, and they didn't use waste out of a trash, but they could in theory. And polyethylene is this plastic that is often used in your single-use plastic packaging, and it's not degradable, so it's gonna be around in your environment for close to 1,000 years. It's comprised of these very long hydrocarbon molecules, and our collaborators run it through a catalytic reaction, which breaks it down into smaller, more digestible pieces. So now this material is a waxy substance. This process essentially pre-digests something that's kind of hard to digest for our microbes to make it a little bit faster for them to do their work. Our microbes don't actually require this step, but they will be much slower without it. So we do a bit of a pre-digestion for them. So then we feed this wax to our engineered microbes. And I'm going to show you some pictures of what you would actually see if you were a grad student in my lab doing this work at benchtop scale. So we have a shake flask right here. We've got some of this wax that came from the waste polyethylene. We add in our liquid broth. That's the broth containing some of our bacteria. And we simply just shake it at about 30 degrees Celsius for up to 72 hours. And we see the broth getting milkier and milkier. That's indication of the cells growing. And as the cells grow, they produce silk, which is our high-value bioplastic. They're actually, and here's a picture, by the way, of what silk looks like, our silk looks like. So these cells are actually converting the atoms in the polyethylene into silk protein, which we can then extract, purify, and use. And it doesn't even have to be silk. We can potentially produce anything that we know the gene sequence of. So that means, imagine turning our waste plastic into insulin, or into another vaccine, or into some life-saving biopharmaceutical. So this platform has the potential to be able to do that. And this is an amazing feat for the bacteria too, because like us, most bacteria prefer sugary foods to be productive. Our strain of bacteria can eat this wax, and that's like saying, give me not the cake, but the candles on the cake, and I'll make for you the Mona Lisa. So where does this is just the beginning. This is a very preliminary proof of concept. But where does this leave us? What is my vision of how this will work in the future, right? This concept of microbially upcycling our recalcitrant waste into useful, sustainable materials. So we're gonna need the chemists to help us figure out how to optimize the feedstock that goes into our cell culture. For example, how do we convert PVC or polystyrene or PFAS into forms that bacteria can easily consume? We're going to need the synthetic biologists to improve the ability of our microbes to consume this very unconventional waste product and to produce products at high rates. For example, bioplastics and biopharmaceuticals. We need our reactor engineers to optimize the reactors. We need our downstream processing purification engineers to make sure that we can purify our product in a cost-efficient way. And we need our material scientists and materials processing engineers to take those materials and turn them into something useful. So this is a daunting task, I think, but it's very worthwhile and exciting. And I think a future where we use nature to make biodegradable plastics using low-value waste streams isn't a sci-fi fantasy. It can be reality, but it needs a lot of work from multidisciplinary, concerted, collaborative efforts. And thankfully, we already have a lot of the foundational technology established. So at this point, I wanted to thank the students in my group, current and past, for making all of this possible and allowing us to really follow our curiosity and do some really interesting stuff. Wow,
I feel old and I feel invigorated. They're gonna take us back in time, tell us how we got here, and then save the world. So this concludes the end of the Red Talks. Please join me in thanking our speakers.